So why don't we go ahead and get started, Eric? I'll pass it to you to introduce Neil.
Sure. I think, boy, Neil barely needs any introduction, but I'll just briefly say that I think he's really pioneered a lot of model systems, and in particular, a lot of the 2D in a dish modeling for KS and a lot of other conditions. But I don't want to just say that because then I'd be minimizing all the work he and his team have done over the years. But he's been one of the foremost experts on KS at the cellular level, and yeah, it's always been great interacting with him, and he's part of our scientific advisory board, and want to thank him for everything he's done. And Neil, I'll let you give a full introduction to yourself. And I think today Neil's going to obviously just talk about one sliver of his work, which is the brain model project. And I'll let you take it away, Neil.
Perfect. Thank you very much, Eric. You can hear me? Just want to make sure
Yep,
you see my slides. Okay,
can
very
hear
good.
you, can see your slides. Everything's working perfectly.
Yeah. No, well, great. I'm happy I can be here today and give an update on brain model. And because brain model is an Dutch national effort to actually understand how we can leverage stem cell-based medicine to understand monogenic neurodevelopmental disorders, including Kleefstra syndrome. And this is is a large effort because, as you will see, there are several elements to it. And I'll just, I think, well, the idea is to give you an update so that you know where we stand. And I think we have done some amazing progress, but there's still a very exciting part that lies ahead of us. Um, and well, I'm happy to share this with you, and also maybe have some discussions at the end on if you have any questions. Um, yeah, so now why brain model? What is the idea of brain model? Well, I think that over the years we have done an amazing job in identifying the genetic causes of neurodevelopmental disorders, and obviously Kleefstra syndrome is a beautiful example of this. We know that it's caused by most of the time a loss of function of the EHMT1 gene. Um, and at the same time, we also have invested a lot in a good clinical phenotyping, natural history of the syndrome. However, and those are essential elements. Knowing what causes disease, we need to know the clinical phenotype. We need to invest in natural history, all in order to at least have an hope and a strategy towards to go to therapy. But for myself, as a biologist, I would say the gap between the gene and its phenotype is a very big gap, and it's something that's very difficult to close, given that typically we have not access to the brain of patients or humans in general. So hence the importance of building brain models, and of course sounds very generic, this name brain model, but it is what the word says. So it's basically develop accessible models, and accessibility I mean with this really at the molecular cellular level, because yes, we can look through MRI scanners at the brain, but we have in order to really understand exactly what's happening, we need to use well, to have a preparation that on one hand harbors the genetics of the disease that we are interested in, and hopefully somehow also reflects a part of the clinical phenotypes that is being seen in the patient. And that's of course a very difficult aspect, and I hope I'll touch upon that and how we want to actually do this in brain model. The core of brain model is really to use stem cell based models, which is, I think now most people are familiar with, but it's based on the fact that stem cells are cell types that allow us to generate any kind of organ. And it's of course from conception on allows to develop an individual. And the stem cells that we are talking about here are actually stem cells that have been re-derived from what we call somatic cells. You can think about blood, hair cells, skin cells, and so on. So we have actually we can take those somatic cells and we can put them back through time almost, and make stem cells from them. And then from that we have stem cells in a culture dish, which we called here induced pluripotent stem cells, and then basically can advance the clock again, let's say, and differentiate the cells in the cell types we are interested in. And in our case, of course, the cells of the that are contained within the brain. Doing so, we can now having our cells of interest in a dish, which are fully accessible at the molecular cellular level, and of course allow us to do experiments on the exactly same genetic background of the original patient, but of course without any kind of harm or disadvantage for the patient, as this is being done in a dish in the lab. And what's more also important to say is that once we have acquired those iPS cells, so these pluripotent stem cells, they are in theory they will grow forever. So we only need once to get some sample from a patient, and once we have the iPS cells, we can keep on culturing and doing experiments with them in the dish, in the lab. Ultimately, of course, the idea is then having the disease model in the dish is in a way also having the patient in a dish, and but of course at in such a way that it's very much reduced. But important is that, or at least the idea is that we can then start not only understand the molecular cellular change, but also try to intervene and change them and potentially rescue the phenotypes. And ultimately, from what we find in the dish, hopefully we can go back to a patient. And that's, of course, a very not an easy feat to do, because that's a very big gap to go back from a dish to patient, given the complexity of a patient and being already a living organism versus some cells in the dish. But I think that the progress in the last years have shown that there is indeed a very important and significant biological signal that is present in a dish that can be useful. And that's what we also want to uncover in this BRAINmodel project. Now, BRAINmodel, those are the aims. It's really to the development of those cell-based systems in 2D and 3D. And there are several elements that are important for that. Is really build robust to increase robustness, complexity, scalability, especially for if we want to do a higher throughput drug testing. But in my opinion, the most important is we need predictability. So how can we increase predictability of what we find in a dish towards patient? We have focused on not on one disorder, but two groups of disorders. On one hand, we have what we called, with the very difficult name, chromatinopathies, and the other synaptopathies. Chromatinopathies are actually genes that are important for the epigenome. That means that it basically, those are enzymes here, some examples on the right, that are putting some mark on the DNA that is then affecting how genes are will be transcribed. And so EHMT1 is one such of a marker, but it actually belongs to a larger family of interesting chromatin remodelers that all have been shown to be affected or lead to a disease. The general concept of BRAINmodel, I think is very innovative, because it has first, I mean, it as I said, it focuses on two types of disorders. They're both neurodevelopment disorders, but I think what it really makes it special is that the idea is not per se to look at groups of disorders, but really taking the individual into account. And then if to do this, it means that we will have a multi-level phenotyping on the individual, on each individual. And the way you can look at this is that we have been taking generating iPS cells from an individual. We have phenotyped this at the cellular level, even looking at single cells almost. We have the same iPS cells have been used to really looking at network activity. So this means that at how thousands of neurons actually interact with each other. And these points 1 and 2 are really what we call the what we do in a dish in the lab. But, and I think this is the innovative aspect, is that we, from the same patient for which we have the cells, we also have acquired real life brain measurements, network activity measurements through the measurements of EEGs. And so basically we put electrodes on the kids' heads, and I'll show this in a second. And so we can measure the activity from these kids, and and having so real measurement of a patient. And then on top of that, we have, of course, the in-depth clinical phenotyping that was done under the guidance of Tjitske Kleefstra. And also important to know is that this clinical assessment also came with the acquirement of biomarkers at the metabolic level, but also immune markers. So this means that at the end of the project, which still runs 2 years, we will have this panel, this multi-level measurements of each individual, and it will allow us to correlate basically all those different levels with each other, rather than making maybe an average of the Kleefstra syndrome patients. We will be able to look at individuals, and I will already show you that indeed we do see, for example, the level EEGs, so very clear differences between patients. Now, when we can have this whole measurements, there's also, of course, some kind of framework in which we like to work, and the framework in which we actually have designed all our experiments is in this framework of this, maybe some of you have heard, this balance between excitation and inhibition. So basically in the brain we have this different cell types, and so this framework of thinking that the balance between excitation and inhibition might disturbed might help us actually design a certain experiment. And so obviously then this we can then use ultimately to with the idea that if this balance is distorted, we can use this as a readout basically at the several levels. So at the cellular level, but also at the EEG level, whether this balance can be restored. And so, yeah, and so ultimate aim is actually to do this for the individual. So it's not per se, at least we want to show the proof of concept that we can do that for the individual, and not per se as a group of disorders, but really tailor our findings to the individual. Now, if we go a bit deeper on this excitation inhibition balance, I think it's an important concept in general for the field of neurodevelopment disorders, but also within our project. So in the brain, of course, consists of hundreds of different cell types, but one can reduce it very basically set to two cell types that determine the output of the activity of the brain, and that's the excitatory cells here in blue and the inhibitory cells in red. And basically those two cells keep each other in balance, so that there is a certain amount of output generated by a cell, the blue cell, so the excitatory cell, that will here show some type of activity. And so when we refer to the excitation inhibition balance, we do not per se mean that there should be as much activity from both cell types, but let's say the overall activity is in balance, and that it's kept in balance because of the amount, or also the intrinsic activity of those two cell types. And so what now, it's almost more than 20 years ago, has been put forward is that in development disorders, this balance might be disturbed. And that of course, disturbing the balance can be in roughly in both directions. one that one has more excitation-driven output or brain. This, for example, can be because there is a stronger excitatory signal, or there is a weaker inhibitory signal. And so you can see here that this all would then lead to more activity. And I think a prime example of this, what I think is very easy to imagine, is what happens during epilepsy or during seizures, is that somehow there is too much activity, a runaway activity, that leads to this hyperexcitability. The opposite is also possible, where there is more inhibition to less excitation, and then the activity would be blunted, basically. And like I said, this is a framework that has been described 20 years ago, and I think there's a lot of evidence supporting this, but it actually has not been so straightforward to measure this kind of balance with stem cells in a dish, nor has it been very easy to also measure this with EEG, so on the patients themselves. And actually, within BRAINmodel, we actually want to also develop tools that allow us to measure this in the dish as well as in the patient. Yeah, so what I want to, one more thing I want to say here is that in this excitation inhibition balance can almost, or our hope would be that at some point it could also be used somehow as a biomarker. Now, how do we measure this in a dish? So we like to call this technology on-a-chip technology. Probably brain is a bit of far-fetched. I mean, at the end, they're just cells, so it's a very reduced system. But what we do is we grow the cells now, those neurons on a chip that you can see here. And what is very interesting is that those neurons will indeed start to show electric activity, as you can see here, with a certain rhythm, and that actually indicates that they have a certain output that is kept in balance between the excitatory and inhibitory cells. And the way we can, of course, then measure this activity, and very similar than the graphs that you saw in the previous slide, you see this kind of activity occurring, and basically when this happens, we can say that the cells have developed. These parameters that you see here, they actually change over time, and they basically give us an idea of how good and how strong the communication is between the nerve cells, but also how this excitation inhibition is. And the analogy that I like to make is that basically we can measure how neurons, nerve cells contacted with each other. So when they are talking to each other, how often, with who, and how loud they are talking. So those are kind of measures that we can extract from these kind of experiments. And what's interesting is that what we see here in the dish, what is highlighted here in gray boxes, this simultaneous activity of the cells, is something that is actually also happening in newborns or in very developing brains. For example, especially just before birth, normally here's an EEG recording of a preterm, and then you see this kind of activity over very different brain regions. If we filter this, then we see that this activity is also synchronized across different brain regions. So it's this synchronized activity is really a feature of the developing brain. So we like to think that at least we somehow can capture some aspect of that, also even in a dish. And so that means that we, on one hand, now can measure this kind of signals in a dish. From the same patient, but ultimately what our goal is to do that from the same patient in the dish, but also relate that then directly to this EEG measurement on this exactly the same patients. And again, in both conditions, we want to look at this excitation inhibition balance. Now, something about EEG then, I already showed you we can do this with the stem cells. So for the EEGs, this part is led by Klaus Linker Hansen and Hilko Beuning. They have developed this kind of measurements originally for cases of very strong epilepsy and also autism. Here you see that, so how it happens in clinic, the kids get a cap over their head. Typically, they get a training cap already so that they can practice at home, so that when they come into the clinic, they are kind of used to wear it. Or we also have there's also they also have a van basically, and they go to the patient's house to do the experiments. And now, this slide here actually shows, it's maybe a bit technical, but it shows basically that they have developed tools to look at this excitation inhibition balance based on the EEG markers. That's of course very important because this excitation inhibition balance, the idea is to measure it from the EEG in the patients, but then also measure that in the dish and make the correlation between both. And maybe this is a little bit technical, but just want to show you here, for example, if you give a zolpidem, that you can change this ratio, that also in idiopathic cases of ASD, so that means where we do not know what the genetic causes is, we see that the variation of this AI balance is much higher than in healthy developing kids. You see, especially the variation can go in both directions. So you here already actually see very nicely that these kind of conditions do not per se lead always to either more excitation or more inhibition, but that the whole range is being actually increased. And then for one particular syndrome, we see here that in this case, at least, we see, yeah, that in general, there is a lower AI ratio. So that means that this brain is more inhibitory driven. But you see, for example, in the individual here, in this case, it has a total normal AI balance, but you also have individuals with very strong skewed AI balance. So what's how far are we? So we have here on the left, you see all the individual patients or genes for which we have recruited patients. So you see the largest group is actually the chromatinopathies, but also the synaptopathies or snaropathies. I just preparing this slide, I realized I did not have the latest numbers. So I apologize. I know that we are now in around 65, 64 patients, if I remember well. So this is not fully up to date. We also see that the most patients are actually from EHMT1, having 9 patients included for which we have EEGs, and for 8 for which we have iPSCs are being generated. So that's definitely the largest represented group. And of course, already for studying a rare disorder, this is already it's a low number, but for rare disorder, it's definitely a high number. Yeah, so that's the current state. So we have finished all the inclusion. Ariana Baumann, PhD student in group from Chitske, has done amazing work in running all those EEGs, but also doing all the clinical assessment. So this is a huge repertoire or a resource, I must say, that we have now. And now we can start doing the work. So especially in the lab, because that's a lot of work to start generating all the cells. And so that's what we are doing with the iPS cells. Just in a little bit what Eric was alluding to, we have been in the lab generating a lot of different cell types in 2D. So we'll not go too much in detail, but over the years we have been looking at the function of the role of EHMT1 in glutamatergic neurons, GABAergic neurons, dopaminergic, serotonergic, astrocytes, and microglia. And below here some overview of the phenotypes we have noticed, we have seen, so some molecular mechanisms. Very recently we have, for example, discovered that the non-neuronal cells like astrocytes play a very important role. But of course, for the excitation inhibition balance, it's essential to look at those two cell types, the glutamatergic and the GABAergic cells, because they are literally the cells that determines this balance between excitation inhibition. But in the lab in general, that's a little bit how we look at it now. So we have all the instructions to generate all the individual cells, and then we basically generate our network of choice, which we now like to have excitatory inhibitory neurons, but then also on top of that, astrocytes and microglia, two important cell types that support neurons, but also help modulating them, and also even in the case of microglia, we think are protective for potential seizure activity. So this is how it looks like. Maybe just go over quickly, but you know, I think it's always very pretty, so I like to show it. The neurons in red here, and then those microglia in green that help sculpt the neurons, I would say, and help mature them. Now, what have we found until now with in general with Clesa patients? So when we put those lines on this microelectrode arrays, very early on already in 2019, we have reported on that. Is that we basically see that the Clesa patient cells do communicate different than the control cells. There's I'll not go too much in detail what all the parameters are, but I think almost visually you can see the difference between the left, which are healthy controls, and the right here, Clesa syndrome patients. The communication is very different. And this example here, I must say, is only with excitatory cells, but when we have done the combination, as you see here, we see that when we even add more inhibitory cells, the phenotype even gets worse. And so our conclusion is a little bit is that the inhibitory cells work less well, and that there is a more excitation-driven direction in the Clesa syndrome patients in the dish. So we can now, as said, we can do that for Clesa syndrome patients, but we can also do that for a larger panel of patients. First of all, you know, we have compared that to several controls, but we have now in the lab also looked at compared the Clesa syndrome with 15 other neurodevelopmental disorders that are more in the metabolic range or channelopathies. And the reason to do that is that actually we can learn also not just by looking at Clesa syndrome itself, but also from other neurodevelopment disorders. So how does it actually is it maybe very similar to another disorder, a syndrome, or is it very different? Because one can of course imagine if there are some molecular if there are some findings being done in another syndrome, and this syndrome might be very similar to Clesa syndrome, that it can be very informative for Clesa syndrome itself. So we've done this for many different disorders, and this is just a snapshot of these networks, how they look like. But it becomes very quickly very complicated, and I just want to show you some piece of data where here in this left here, every dot is basically a cell line and a patient and a recording. And so what we can do is we can try to make this very complex data set much simpler. And then what we can then do is for every cell line actually compute how close to another cell line they actually behave. And so what is very interesting, if you do this, you start to see those kind of clusters basically by those dark blue kind of squares. And that basically indicates that certain cell lines behave very similar. And I think it's very interesting to see that, for example, we see here the clustering of the EHMT1 lines together, but also together with other disorders, for example, with patients with a mutation in SMARCB1. And that's interesting because we know that those patients have also an Kleefstra kind of like syndrome. So it's another syndrome, but it's relatively close to Kleefstra syndrome. So, and it's kind of encouraging to see that we even see that in a dish, we can actually somehow see that the phenotypes are very close to each other. Further, we can learn how variable this kind of measurements are for each individual patient. So, for example, here we have asked the question from all the cells we have for each of these syndromes, how often do they cluster in one of these colored clusters? And then you see that EHMT1 patient lines always fall in the orange or in the green cluster without exception. And again, shows that on one hand, there is variability amongst the lines, so maybe also amongst the patients, but this is exactly what we want to leverage. We want to really leverage the individual patient's data and not see it as one average. Good. So then to move on, so then ultimately what we want to do is then connect our in vitro data with our EEG data. And so I showed you some in vitro data for HMT1. So what about the EEG data? So first of all, disclaimer: I will not give you unfortunately the whole data set analyzed. So it's basically full analysis now because it's a quite complex data set and there are many choices to be made. But I asked the student that is working on this to give me a high level data on the EEG. And so what you see here is a plot where they looked at severity of the EEG abnormalities based on different gradations, and then for the different chromatinopathy patients for which we have the EEG. And then I think what is interesting is that, first of all, the EHMT1 patient seems to be more on the severe case. So that means that they have, you see EHMT1, several of the patients here, most on the left. There's only two EHMT1 patients that had actually a fairly normal EEG. And this already shows the huge variability within the patients. We have a very severely affected patient at EEG level, and we seem to also have patients that is rather normal. It's in certain EEG parameters. So, and this of course I think is something that we all also know from the clinic, that patients can be very different in their clinical manifestation, but this is also seen at the EEG level. And of course, our question is can we also see this now, for example, in a dish in our model system? Some analysis that was done, and again, is very preliminary. So let's say take it with a grain of salt. Is so when we record EEGs, there are basically thousands of data points, and then it's always difficult how to analyze this. And in this first analysis, basically they reduced all the data, and the patients were actually compared to age-matched and gender-matched controls. And then what you see here in the gray square is actually the space of the phenotypes that is being taken by all the typical developing controls. And then the colored circles or symbols here are where the individual patients actually are being distributed. So I have to correct myself. So the gray squares are all the controls, and the crosses here, those are actually the age matched to the Kleefstra patients. So I think what's interesting to see is that that indeed patients do deviate from each other. But then we also have this one patient here that seems to be in the middle of the controls, and that's probably the same patient that I just showed you before. We can do that at different levels of analysis, and you see that actually quite consistently that EHMT1 patients do show an abnormal EEG. For the details, they show this increased low frequency power and high frequency DFA, and also asymmetry between the activity. So there's more an anterior shifted symmetry and also asymmetry between the two hemispheres. So that means that some of the activity is more anterior-wise, and there is less symmetry between the left and the right part of the brain. And it's interesting because it is that those are actually features that have been seen in more in the general population of neurodevelopmental disorders, and does reflect in a way an atypical brain brain network maturation, but and also reduced processing speed. Although we have not yet measured this AI balance here, the data shown plotted on the right are actually suggestive also from a difference in excitation inhibition balance. But this analysis is ongoing. So how do we now actually link now this EG data to what we find in the dish? So that is also ongoing. So we now have all the data. We just want to show you we have actually made progress in the in silico modeling of how we want to do this. So as we start with patients, I've shown you we have MEA recordings. We also have EG recordings, but those are two different worlds and two different types of recordings. So how do we bring this together? Well, we do this by also not me, but the people in the BRAINmodel consortium. They built an what we call an in silico model. So it's a computer model that can actually predict or how to say duplicate or mimic the signal that we measure. And because this model, we can then change all the parameters, we can actually probably try to simulate the real measurements. And that works fantastically well. We have published on this. So it means that now when we have the real measurement, we can actually tweak the parameters such a way that we can mimic that. But by tweaking the parameters, we know actually what is wrong at the cellular level. And now we can switch the model to an EG model that does exactly the same. And then we can maintain the parameters that are changed. And now we can apply them to an EEG simulation. And then we do the same. So basically we would have done an EEG from the patient. We have the EEG simulation. And again, here there are some parameters that actually helps us going from the simulation to the real recording. And in that way, we can then actually going from one modality, the EEGs, to the MEA, or from the MEA to the EEGs. I think it's very novel to do it like this, and it is still ongoing, but it's very exciting that we can now almost start doing this. Okay, so the last minutes or so, I want to maybe go also on how we now also want to use this information for strategies to therapies. Basically, we have actually two strategies. One is to actually look at, can we actually interfere with this balance by doing drug repurposing, or can we also leverage antisense oligonucleotide strategies? In order to come to compounds, what we have developed, and I think that's also a very innovative methodology, is actually to now also link our measurement in a dish to the transcriptome change, so the changes in gene expression in that same experiment. And what it allows us to do is actually to show that indeed the molecular changes, so at the gene expression level, actually are very predictive to the activity changes, as we see that the grouping is very similar between the transcriptome data and the activity data. Now, this might sound very complex, but what we can do now is we can see at the activity level indeed that certain cell lines group together. And again, I think what's very exciting to see is that the Kleefstra cells group here together in this cluster, but together with the Kleefstra syndrome-like cells. So again, showing that probably this really makes sense. But again, how do we use this now to find some kind of lead compounds? Well, what we do is we try to identify the genes that for example, are changed in Kleefstra syndrome. And here is just hypothetical. I call them genes A, B, C, and let's assume they are upregulated. So there's more expressed in the Kleefstra syndrome lines. We then look in this links database, which is a large database with 20,000 compounds, where they have also looked at the effect of compounds on these genes. And then we are looking for compound that actually does exactly the opposite on these genes that are upregulated. So if in claves are they upregulated, we look for compounds in which that induces downregulation. So basically to balance out the gene transcription. It's of course a bit more complicated than illustrated here. But so the question is does our data actually show that we can do that? And I just want to show a proof concept. It's not with EHMT1. I apologize for that, but I think that the proof concept is very interesting because we basically, in this case, we ask the question, can we now use this kind of approach, for example, to go from one cell state to the other, so in this case, from control cells to another disorder. And we selected 5 compounds, different concentrations. And the way we do these experiments is that we ask the question, can we now, and it's really proof concept, so can we now go from a control condition to a disease condition using our algorithm and our approach? And so what is interesting to see, so here you have all the controls. So if you start adding certain compounds, you see that there is this beautiful time and dosage dependent movement from this cell to the control to the disease. And so this is very early after, so only 1 day after adding the compound, but then after 2 weeks adding the compound, this cell is now completely behaves like a disease cells. And so we can do that for different drugs. And this really shows that we now have this a tool that allows to look at concentration time dependent modulation of the network. So it's really a very nice screening platform. And so we will use this now also for the AHMT1 patients, or at least that's what we hope. Okay, then really last part is then can we now so for the antisense oligonucleotide. I wanted to put this in. This is actually not really part of BRAINmodel, but from another consortium. Because I know that in the whole community of rare disorders, there's a lot of attention or a lot of, let's maybe hype even, about these antisense oligonucleotides, because there seem to be some successes. And the question is, of course, would this also be applicable for Kleefstra syndrome? And so we recently published a paper where we actually did more the theoretical analysis as to ask the question as for how many neurodevelopmental disorder conditions would actually be any type of antisense oligonucleotide strategy be applicable. And so here we see actually, so we went to a large data set of all the disorders, all the individual mutations, and so we selected a group of intervention that are based on AONs, and then we looked at how many would actually be amenable for therapy. And then we made some criteria that at least there should be 2 independent criteria. And our number comes down to about 1/3 of all the neurodevelopment disorders could theoretically be tackled with AON. So this was actually quite a large number. But of course, mind is this pure theoretically. And one approach that I think has gotten a lot of attention is the approach we called Tango, which you might have heard, is where we actually try to increase the expression of a healthy variant. And in order to, at least this seems quite prominent, interesting approach and effective in Dravet syndrome. I'll show you that in a second. So we really wanted to investigate, could that be also for EHMT1 or other neurodevelopment disorders be a strategy? And just to illustrate how actually Tango works, I illustrated for this genes SCN1A, which is now in clinical trial. So, but you could replace SCN1A, for example, with EHMT1. So we have 2 healthy controls. This could be seen as the 2 faucets. You can fill your beaker with water. In the disease situation, you have one allele, one chromosome that is not working. So you have basically less water coming out of the faucet. And so the way is not to repair the broken one, but actually make the other one work harder. And this is done by using AONs that basically target a certain exon that normally would lead to stop codon. And by using this AON, you would skip this exon, allowing the gene to be expressed. And in this case, there would be actually more expression of functional mRNA. And this is actually, in order to investigate whether this is also possible for NDD genes, we actually need the information of this exon inclusion-exclusion. And so this is something that we have done together with a student of mine. And we have done this in different cell types, all iPS-derived lines. And we have actually looked at all these events that happened, and basically to have a good idea whether this Tango approach would be, for example, a good approach for HMT1. We are working on a manuscript and also to release all the data so that actually everybody can look for it. We will make it available online also with the database, and this is a bit how it will look like. So this for EHMT1, for example, we have here the expression of EHMT1 in iPS cells, but also in the different cell types over time. So we see, for example, that in neurons, EHMT1 is relatively low expressed. It's actually more expressed in astrocytes and microglia. And then also there will be actually tools to look at all the different kind of exon inclusion, exclusion, allowing also to look whether an Tango approach might be beneficial, might be possible for HMT1. So I hope this will be very useful for everyone. Bottom line, I think there is little evidence that genetically, at least, that a Tango approach would be beneficial for EHMT1, as the percentage of this kind of special exons seems to be very low, but maybe we should look more in depth for this. With this, I would like to end. Thank all the BRAINmodel people. We had a conference last Friday. We had the luck of having Sergio Pasca visiting us for giving a keynote. Yeah, with this, I think I probably over my time. I'm apologizing for that, but I'm still happy to take questions.
Thanks, Nael. That was amazing. It's so exciting to see all this going on. I think maybe we should do just one question because we are running behind time. So there's a question. I am wondering about the EEG data where you see some KS patients have more severe deficits on EEG compared with others. Do you see this clustering based on variant type?
That's a very good question. That's of course the first thing we did, and that's easy to check, but that's not the case. So we don't see that. No, we don't. But of course, it's always low numbers, right? So it's that's always the biggest problem, because you can ask the same question: do you see clean? You don't need an EEG for that. You also classify it, clinically. It doesn't correlate with the variants, not. They also. No, and I think no, we did not see that. Short answer.
Okay.
But we do hope to see, of course, whether it will correlate to our in vitro data. That's of course something that we hope. Oh, hope. Well, that's the question. So I mean, I don't know if I need to hope that, but it's I think one of our main questions.
Okay, great. So yeah, we'll need to move on to the next speaker. I have a bunch of questions for you, Nael, but I'll send them to you offline.
Yeah, please. And I can just tell I'm happy to answer questions offline and via email. So please, happy to do so.
Yeah.
Thank
Thanks a lot,
you,
Nael.
and I apologize for running out.
No worries.